Do you know what a virus variant is? Do you know what an isolate or a strain is? If you don't and you'd like to find out, keep on watching. I'm Vincent Racaniello, and I've been a virologist for nearly 40 years. And today I want to explain to you the terms virus isolate, variant, strain, and a couple more. As you're well aware, this is March 1st, 2021. Pandemic has been on for about a year now, caused by SARS-CoV-2. Many people are thinking about viruses, and so they're using terms that they may not be familiar with, like isolate, variant, and strain. So I thought I would tell you what I think those terms mean. Now, the problem is, as shown in this first slide, there's no universally accepted definition for these terms, strain, variant, and isolate, in the virology community. So how can we expect that everyone else gets them right? What happens is you're trained in someone's laboratory and you hear them say things and you associate a meaning with them, and that's what you do. And they may or may not be right. By the way, this little nugget of wisdom comes from this paper where Jens Kuhn is the first author. It was published in Archives of Virology in 2013. And I will make a few other quotations from that article as well. Now, despite this looseness of definitions, for example, there's no one place where you could go and find the definition for variant, for example, or isolate or strain. I looked in several textbooks. In fact, not even my textbook, Principles of Virology, co-authored with 4 other virologists. Not even there will you find a definition of these terms. I think we need to put it in the next edition, though. Let's go through and try and define them and put them in some kind of context with the current outbreak. Let's start with isolate. What is an isolate? An isolate is a virus that we have isolated from an infected host, and we have propagated that in culture. Now, isolating encapsulates the meaning. It means that you take a sample from someone who you think might have a virus infection, you put that sample in cells in culture, which is how we grow viruses, and you grow a virus, and now you have an isolate. Let me illustrate that for you here. So we do a nasopharyngeal swab for, we suspect someone might have COVID-19. You take the swab, you put it in a tube, which has a little fluid in the bottom. It's brought to a laboratory. And in the laboratory, you take a little of that fluid and you add it to cells that are growing in a culture dish. That is how we isolate viruses. And if that person had a virus in him or her, it would grow up in culture, and then you could take the medium from these cells and store them in little tubes, which is what we do in the laboratory. So that's what we mean by isolate. We have actually isolated the virus. Now, most of the time, we take this nasopharyngeal swab in the solution and we just do the genome sequence. And we don't actually have a physical isolate virus. And that's very important because an isolate means you have the virus in a tube in a freezer somewhere, not just the genome sequence. The genome sequence is representative of the isolate, but it is not an isolate. So we have hundreds of thousands of genome sequences from SARS-CoV-2, but not that many virus isolates. People get the sequence, they work with the sequence, they don't necessarily work with the virus. A genome sequence represents an isolate, but it is not an isolate. And as I said, there are hundreds of thousands of full genome sequences of SARS-CoV-2, but most are not isolates. The virus was not isolated in cell culture. Now, we give isolates names to keep track of them. I have hundreds of thousands of genome sequences representing isolates or isolates. We want to know a few things about them, like where they came from. And so here are some examples. This is a phylogenetic tree of about 4,000 or so genome sequences of SARS-CoV-2 isolates from various people from 2020 through the current time. And you can find this at nextstrain.org. Great resource, by the way. Go over there and just poke around. You can learn a lot by doing that. This phylogenetic tree, what we've done here is to take all the genome sequences and compare them and say which ones are most closely related. And so you can make a phylogenetic tree of those. And you can click on any of these dots. The dots are individual genome sequences, which may or may not have an isolate associated with them. And so here, for example, one of these first isolates back in 2019 is called Wuhan/HU1 2019, Wuhan human-1, and it tells you the date of isolation, December 26, 2019. And the clade number, we'll get to that a bit later. That's not actually the correct way to name these. This shown on the right here is actually how you would name a SARS-CoV-2 isolate. SARS-CoV-2, by the way, is the virus, and then we give the isolates different names so we can keep track of them. Beta coronavirus, it's the genus in which SARS-CoV-2 is located. Wuhan is where it was isolated from the patient, typically an isolate number. Here it's WIV4, which means Wuhan Institute of Virology number 4, and then the year of isolation. You can get a lot of information just from looking at that, right? That's really important. Then down here is another way that we name isolates of influenza viruses. Every virus has their own way of being named in isolates. This was influenza A virus A. There are 3 types, A, B, and C. A/duck. So the second term means what kind of host it came from. The third is the geographical location. The fourth is an isolate number. The fifth is the year, 1968. And then the subtype in terms of the hemagglutinin and neuraminidase, H6N1 in this case. That's great. I love the influenza virus nomenclature. They do a great job telling you an awful lot about the virus. All right, so every virus isolate is named in this way. What about variants? What is a variant now? We've got isolate down. Let's look at variant. A variant is a virus whose genome sequence differs from that of some reference virus. So in the case of SARS-CoV-2, the very first isolate that we had from Wuhan in 2019, everything else that we isolate from then, I'm using isolate in the colloquial sense because we mostly have genome sequences, right? Everything derived from that original would be potentially a variant. So that's what we mean by the genome sequence differs from that of a reference virus. Really important point about variants. No inference is made about whether the change in the genome sequence that you see causes any change in the phenotype of the virus. The phenotype is how the virus behaves. For example, I shouldn't say behave, that's anthropomorphizing, but the properties. For example, the physical stability of the virus, which kind of hosts it can infect, what kind of disease it causes. Those are all phenotypes of the virus. There are many, many different phenotypes, which of course are controlled in part by genotype. So when you say variant, you make no inference about what these changes are doing for the virus. You're just saying, here is a virus that differs in genome sequence from my original isolate. So let's take a look at this now on the context of this nextstrain.org classification. So again, we have our Wuhan number 1 way back in 2019, which I showed you before. Now I clicked on one of these blue dots downstream of it in the phylogenetic tree. And you can see here it's got an isolate name, Bulgaria 15 2020. That's great. We know it came from Bulgaria. It's given an isolate number 15 in 2020. And in fact, May 13th specifically. And you can see it has these genome changes compared to the Wuhan virus. It has 3 nucleotide mutations from a G to a T at 16,917 base number in the 30,000 base genome. And then amino acid changes, 2 of them, as you can see here. This is not correct to call them amino acid mutations. A mutation is a change in nucleotide sequence. That is very much established. So whoever decided to make this site is wrong about that. I'm sorry. So you can click on any of these dots and see the changes with respect to the ancestral virus. Now, an interesting question is every of these isolates a variant? Because if it has a different genome sequence, it may differ by 1 or 10 or even more nucleotide mutations. Are they variants? I think the answer is yes. The problem is that genome sequencing at this scale is pretty new. And, in the old days meaning before the time we were able to sequence isolates by the hundreds of thousands, variants were more rare because we didn't do much genome sequencing. And so when we found one, we said, yeah, it's a variant. Now almost every one of these isolates on this next strain map is likely a variant. Not every one is different. I could go over some of these dots and you'd see that some of them are identical, but every one of these is different, or nearly every one, so most of them are variants. I'm comfortable with saying that every different SARS-CoV-2 isolate, we're inferring nothing about what those changes do, what those mutations do. Some of them may do nothing, and some of them may have some phenotypic effect on the virus, but I think this should all be called variants. I'm sure you've heard of the term variants of concern, and this arose earlier this year when a new variant was detected in the United Kingdom, and that's described in this technical briefing here, which you can go look up, the title of which is Detection of an Epidemiological Cluster Associated with a New Variant of Concern. So I took this part out for you to see because you can see that they're actually using the nomenclature correctly. And they say nomenclature of variants in the UK. SARS-CoV-2 variants, if considered to have concerning epidemiological, immunological, or pathogenic properties, are raised for formal investigation. At this point, they are designated variant under investigation. That first sentence is really important. These variants, if they have different properties, concerning properties in this case, we investigate them, but we're still calling them variants. If we don't think they have any different properties, we're still going to call them variants. We just won't investigate them. Then they could be designated variant of concern because there's some risk assessment done which says that we're a little worried about these, and that's where that term came from. Now, so they understand that a variant is a virus with a change, as I've told you. And I really appreciate what the UK did, the UK scientists did in this case, because they called them variants. And many other people were throwing about the term strain, which, as you'll see in a moment, is not correct to describe these. So they're variants, and that makes it necessary for the press to call them variants because they're variants of concerns. They're not strains of concerns. They're variants of concerns. Okay. Now, what is a strain? A virus strain is a variant that has unique and stable phenotypic characteristics. So you know what phenotype is. It's some property that may result from a genetic change, right? And we're saying here that a variant strain is a variant with unique and stable phenotypic characteristics. Unique meaning we've never seen these before. And so the UK scientists call their SARS-CoV-2 viruses isolates variants. They don't call them strains. So when do you call them a strain? Well, that is the key. Not everyone should be able to call it a strain. A journalist describing this should not call it a strain because he or she is not an authority. And you look down here, I put on this slide another quote from that paper I showed earlier where Jens Kuhn is the first author. The designation of a virus variant as a virus strain would be the responsibility of international expert groups. What does that mean? There are groups charged with virus nomenclature. In fact, the International Committee for the Taxonomy of Viruses, of which Jens Kuhn is a member, decides these issues, what to call viruses. And they would look at all the data at why you think this variant has unique and stable phenotypic characteristics. And then you might call it a strain. But until then, you shouldn't. Why not? It's confusing. If some people call it a strain and others call it a variant, you shouldn't. You should use common nomenclature. And so the you see the hierarchy here: isolate, variant, and strain. It's very clear. You do some experiments in the laboratory, in experimental animals, you make observations in people, you put all these together and you say, we have something unique here. We think this merits the appellation strain. As far as I'm concerned, so far, there's one strain of SARS-CoV-2. That could change. For example, last year, a virus with a single amino acid change in the spike, D614G, arose in February, and now pretty much every isolate has that change. So it propagated throughout the world. Clearly has some fitness advantage that allowed it to propagate globally. And when that's figured out, it could get the designation strain. It would be distinct from the original isolate, which has the D at 614, not the G. But it's not been done yet. You shouldn't call it a strain because Jens Kuhn and his colleagues have not said that D614G is part of a strain. Now, you may be asking, what else would make a strain? Well, there are the virus I work with, poliovirus. There are 3 serotypes, which you could call 3 strains because they have unique phenotypic characteristics. They react very differently with antibodies. And by the way, types 2 and 3 have been declared eradicated. So only type 1 is circulating now. So there are these 3 serotypes, and I'll explain serotype in a moment. Then there are mouse-adapted polioviruses. So people have taken type 1 and type 2 and type 3 strains and passaged them in mice, and then they acquire the ability to cause disease in mice. And those are strains because they have unique characteristic, unique being able to reproduce in mice because the human viruses, polioviruses, do not. And they're stable. They don't go away. So there are multiple strains of poliovirus by that criteria. And that's what you would need in order to say that a virus variant is a strain, in fact. Now, I mentioned this word serotype. What does that mean? So viruses of the same species, for example, poliovirus, that are antigenically distinct. And I gave an example before, the 3 serotypes of poliovirus. The vaccines that we use to prevent poliomyelitis, there are 2 different kinds of vaccines. There's the Salk vaccine and the Sabin vaccine. They're made very differently. Each of those at one point had 3 serotypes in it. It was a trivalent vaccine because all 3 serotypes were circulating. If you were infected with one, you were protected against it. You never got infected with, say, type 1 again, but you could be infected with types 2 and 3. So they were antigenically distinct, and the antibodies induced by one would not block infection with the other or block disease caused by the other. So that's what a serotype is. It is viruses of the same species that are antigenically distinct. We have 3 serotypes of poliovirus. Now, historically, serotype has been defined by what we call neutralization assays, and I'm showing that to you here on this slide. These are cell culture dishes that have been stained, and these are plaques. These clearings on each one is a plaque, and this is a plaque assay where we can measure virus infectivity. And I have a video which I'll link to above where you can see how a plaque assay works. But basically each clearing or plaque is caused by an infectious virus particle. Now on the bottom, what we've done is we take, I don't know how many plaques are here, 20 plaque-forming units, and you can see each of these plates has the same number of plaques because we have mixed these viruses with a saline as a control. On the top, though, we have mixed the viruses with different dilutions of serum taken from an animal that had been infected with poliovirus, say, or from a human that had been immunized with poliovirus vaccine. And here the dilution of 1 to 10, that's the dilution of serum. So it's fairly concentrated. You can see there are no more plaques. You mix the serum with the virus, you incubate it for a short time, then you do the plaque assay. You see there's no infectivity. There are no plaques left. 1 to 100, there are no plaques left. However, at 1 to 1,000 dilution, now you're diluting out the antibodies. You now get 2 plaques. And then at 1 to 10,000, no more neutralizing activity. That's a neutralization assay. And by the way, you would say this serum has a titer of 1 to 100, the last dilution that gave you a blocking of infectivity. So if you do this with type 1 poliovirus, let's say you use type 1 poliovirus as the virus in this experiment, and you have serum from an animal that had been immunized with type 2 poliovirus, you would see no neutralization. It's a different serotype. By looking at the genome sequence, you're actually looking at the genotype, and that's what I want to explain here. It's another term for describing viruses. Genotype, a description of the genetic makeup of the virus. Essentially, you do the genome sequence and see if the viruses fall into different genotypes. Now, we do this for some viruses and not others. For example, hepatitis C virus are classified into 6 genotypes. So if you do a genome sequence, you can immediately say this is genotype 1 or 3 or 6, whatever it may be, just by looking at the genome sequence. There's a certain percent identity associated with being a genotype. And the hepatitis C viruses are further classified into subtypes like 1A and 1B. So that's genotype, the genetic makeup. And you can look at the genotype now and predict for many viruses the serotype. There's another term you will see called clade. And if you look closely at the nextstrain.org pictures that I've shown you, you see clade on it. And a clade is a group of viruses composed of an ancestor and its descendants. Okay? An ancestor and its descendants. And you can see on this image, I have 4 different phylogenetic trees created from viral genome sequences, say. And in green, that's a clade. All these points or tips of the branches are descended from one ancestor. There's another clade here in blue on the right. That one is descended from an ancestor. But clades don't extend across branches of the tree. So on the lower left, that's not a clade. And on the lower right, that is not a clade. So that's defining clade as an ancestor with its descendants. So let's take a look at that in the next strain phylogeny. So here's, again, about 4,000 sequences. There are many, many, many thousands of more, but the computer program can't handle it. It gets very inefficient and breaks. And you can see on the upper left, it says phylogeny clade. You have a whole bunch of clades here, and they're colored differently. 19AB, 20AB, 20CD. And you can see they correspond to the colors on this phylogenetic tree. And so on the actual website, you can say click on the 19A color, and it will illustrate all the members of the 19A clade. And again, it consists of the descendants of a common ancestor. So here we have in the very dark blue, that's the first clade, 19A, descended from the original Wuhan isolate. And since then, because the virus has been reproducing in so many people, it's diversifying. You have many different clades being established, and you can see them on these colors here. They make perfect sense in terms of the definition. That is the ancestor and all its descendants. And that's what a clade is. And many other viruses, the term clade is used. Sometimes it's not used for viruses. It really depends. It's a relatively new term, which is empowered by genome sequencing. Before that, we had no way to make such a finely tuned phylogenetic tree because we didn't have genome sequences. That's isolate, variant, and strain. I think those are the right definitions. They make sense. I hope you understand it. If you have questions, put them down below. And let me leave you with the thought or the question, what about the SARS-CoV-2 variants? Should I worry about them? And you may hear every day in the press about a new variant that's been detected and that it's going to be the end of the world. I think you should be careful about that. And that's why I'm leaving you with this question. Are they variants or scariants? This is now a term that is propagating through the scientific community because the media, of course, likes to scare you. That's how they get your attention. But not all these variants are scary. And in a subsequent video, I will give you my thoughts on whether all these variants are scary or not. And the bottom line is not all of them. The real issue is whether the changes in these variants avoid the immunity produced by infection or vaccination. And as I sit here today on March 1st, so far, it looks like most of the vaccines that we have are taking care of the variants. And that may change, of course. But I will talk about that in a subsequent video. I'm Vincent Racaniello, and I'm Earth's virology professor. Thanks for watching.